Hello, students, and welcome to my screencast on photosynthesis to help you for your upcoming assessment. Here you see a beautiful picture of our TASM pitch, right? And photosynthesis going on there. And don't forget our friend, our chloroplast model here. So just like all my videos, this is meant to help you. It's not meant for commercial gain or profit. All the images, I try to be more careful this time, are from Google Images and with a free to use license tag, order from Khan Academy, and you can access Khan Academy at khanacademy.org. So let's jump in. Why is photosynthesis vital to us? So hopefully you can come up with the photosynthesis equation, right? Autotrophs, so some bacteria and plants can take carbon dioxide and water and energy from sunlight or maybe from hydrothermal vents, and they're able to turn that into glucose and oxygen. That glucose they can then break down themselves in the process of cellular respiration to make ATP for themselves. So how cool would it be if we could do photosynthesis, right? We could just, instead of lining up at Crafty Kitchen for lunch, we could just go outside to the soccer pitch and enjoy some sun. But unfortunately, you would just get sunburn. So hopefully you could go through this and practice with the numbers, and you can see that it's a balanced equation. All right. How do plants get the reactants they need for photosynthesis and release the products? Can you label the parts of a leaf on the inside? And this comes from your POGL that we went through and from doing the lab in class. Go ahead and pause the video. All right. So, right, plants are going to have roots. They're going to be able to get water through their roots from differences in water potential. And then inside the leaf, we had a cool model that we looked at in class. So here would be like the palisade mesophyll. This would be the upper top part, the cuticle. And here are the stomata, or the holes. And these stomata are where oxygen and carbon dioxide can be exchanged. So oxygen would leave and carbon dioxide would come in. These cells have lots of chloroplasts lined up for optimal photosynthesis. So in our fun lab that we did, we took cutouts of a leaf, or little chads, you could call them, and we hole punched them. And we sucked the air out of them using a syringe, filled it with water, and it became more dense than regular water and sunk to the bottom of a beaker. And we added some baking soda as a carbon source. And as the leaf parts did photosynthesis, they actually produced oxygen. And this rose up in the beaker, which is pretty fun. And the oxygen, you can see it coming out of the stomata. Here, it's been a while since we've done this, but remember that this would be a chloroplast. The little ring-like structures inside that look like green pancakes are called thylakoids. A stack of them is called granum. And then the fluid on the inside that has special enzymes in it is called stroma. All right, that's pretty cool. A lot about plants right there. Let's dig into the process of photosynthesis. I'm going to highly encourage you to take the papers I gave you in class and to model this. Okay, can you remember, why are plants green? Okay, sorry for that pause. Just had to tell some people in the hallway to be quiet. So let's do this. So why are plants green? All right, so hopefully you remember that they reflect green light. So here, many of you did this for your virtual lab, and you showed that they're really able to absorb blue and red light. And then they reflect green. So when you put the green light on the plant, it did not do a lot of photosynthesis. So that's cool. Okay, here is a model of what's going on in photosynthesis. And so can you try and label what each of these little pieces are here? Let's go through them together. So number one is the stroma. That is the liquid inside the chloroplast that has enzymes in it. Number two is going to be the thylakoid. It's the green-shaped pancake where the light-dependent reactions are going to take place. Number three will be water coming into the system. Number four will be oxygen leaving the system. So light hits this. There's going to be a reaction here, and we'll get into that in the next slide, but water is going to be split in half, and then oxygen is going to be able to leave. All right, this is going to produce the light-dependent reactions. They're going to make the reactants for the Calvin cycle. The Calvin cycle is going to make the reactants for the light-dependent reactions. So you can see a big cycle here in between what we call the light-dependent reactions and the Calvin cycle. So the light-dependent reactions are going to produce ATP and NADPH. This would be energy and a high-energy electron carrier. They go to the Calvin cycle. So that's the Calvin cycle is going to take CO2. It's going to fix it to a pre-existing molecule, and then it's going to reduce it, and it's going to become a sugar, or G3P. This is going to take energy and the electrons from NADPH, so it's going to send back used up energy, or ADP, and the empty taxi cab NADP+. All right, let's roll through those light-dependent reactions. So pause the video, use a word bank, see if you can figure out what's going on again here. So number one, this would be the stroma on the outside. Number two, this would be the thylakoid, or the inside the thylakoid here. So light's going to hit this membrane, and it's going to hit it all over, right? But as it hits these certain spots called photosystems, it's going to hit a specific molecule called chlorophyll. That chlorophyll is going to get excited, and the electrons will go through as it gets excited, the chlorophyll, it's going to lose an electron. And when it loses those electrons, it's going to go through an electron transport chain. The electron transport chain is going to pump hydrogen protons to the inside of the thylakoid. So light's hitting right here. It pumps the H pluses into the thylakoid, into it. So you might say, well, where do those come from? It comes from splitting water to replace the electrons that were excited in chlorophyll. And so the electrons are going to be taken from the H's. If you remember, H has one proton and one electron. So when it takes the electron, all that's left is a proton, or an H plus. So those H pluses are in here. Now, some of them got out here into the stroma area by diffusing through this pump called ATP synthase. So let's go on to that. So number four, the electrons, they go through the electron transport chain. They're going to get into a taxi cab, is what I call them, or NADP+. NADP+ is an electron acceptor. It's going to carry them. It's going to turn into NADPH. NADP+ plus two electrons. The electron gets rid of the plus an H plus makes NADPH. All right. As the H pluses here, there's more of them on the inside of the thylakoid than on the outside. So it can diffuse through ATP synthase. And just like in cellular respiration, when it diffuses through ATP synthase, it's going to turn the rotor. That's going to phosphorylate an ADP to make ATP. So it's going to take a phosphate, it's going to take ADP, and it's going to push them together to make a charged battery. So that is pretty cool. So why can't plants just exist on the light-dependent reactions? Why do they have to also have mitochondria? Why do they have to break down sugars? Why can't they just get enough energy from here? All right. So hopefully you thought about that, and hopefully you think that, well, not all parts of a plant are photosynthetic, right? The roots are not photosynthetic. The flower might not be as well. And so they're going to need to break down sugars. Here is another picture of it, just so you can see a different model of it. So here would be, this would be the thylakoid, right? So this would be the green pancake right here. Light's going to hit our green pancake. It's going to excite the chlorophyll. The chlorophyll is going to lose electrons. It can't just sit there with lost electrons. It's going to replace them. And to replace them, it does it by splitting water. And it steals the electrons from the H's. Those H's become H pluses because they've lost their electrons. There's many of them here on the inside. They're able to diffuse through ATP synthase, and that's going to make ATP. So if you're really astute and if you really caught on to it, you might be thinking, well, eventually, Mr. Gibney, there will be as many H pluses in the stroma or in the liquid out here as there are on the inside of the thylakoid. You're right. And so what happens, or what the thylakoid does, is that as the electron goes through this electron transport chain, it's giving energy to these protein channels to pump H pluses back in. So it's using that energy to move the H pluses against their concentration gradient. So from an area of low to high H pluses. That way, the high H plus gradient on the inside, or the high H plus concentration on the inside, can diffuse out through ATP synthase. So what is the big picture here? The big picture is that we made NADPH and ATP. Through this process, we had to have water. That water was broken down, and it releases oxygen into the atmosphere, which gave us the oxygen we can breathe. I would encourage you to go to page 414 in your book and to do the animation in this with your eyeball and to challenge yourself to model. All right, so here's the big picture again. The sunlight came down. It's going to hit all these thylakoids. Notice how there's a whole bunch of them, so this can be going on a whole lot. When it hits it, it's going to hit the photosystem. It's going to lose those electrons. That's going to split this water molecule. Splitting the water molecule releases oxygen. The electrons are going to go fill up NADP plus, and they're going to also take an H plus, and they're going to make NADPH. The H pluses are going to diffuse through ATP synthase to make ATP. So the light-dependent reactions make two things that the Calvin cycle needs. They're going to make ATP and NADPH. The Calvin cycle takes place in the stroma, this liquid out here. So there's lots of enzymes that can catalyze. These enzymes are going to take ATP and NADPH and CO2 to make a sugar. All right, and then they'll return the used up goods. They're going to return the used up energy. They're going to return a dead battery in the form of ADP, and a used up taxicab in the form of NADP+. All right, so can you describe the three main stages of the Calvin cycle? Fixation, reduction, and regeneration. You do not need to know any of the enzymes. You don't need to know any of these numbers. Just want you to try and get an appreciation of what's going on in the stroma here. So in fixation, the carbon dioxide is attached to a pre-existing molecule. There's this enzyme called Rubisco. You don't need to know it. It's probably the most prevalent enzyme in the world, though, and it's going to attach CO2 to this molecule. It's then going to split, and with these ATP and NADPH that you made in the light reactions, that the thylakoid made, it's going to generate sugars. Right? So it's going to take all the electrons from NADPH and phosphates, and it's going to make G3P, which can make glucose. So you're sending back the used up energy and the used up taxicabs. This pre-existing molecule needs to be remade or regenerated, and that's going to happen in regeneration. So that's all you need to think about here, is that it's going to remake the starting molecule. All right, and so you can go through my slides and see that. And then this one, I'm going to really rely upon your lab to do it, okay? I'm not going to have a bunch of slides on it, but what are some factors that affected photosynthesis? And you should feel comfortable with each of these. And the next class, you'll get to practice them again. All right, and so I hope you enjoyed photosynthesis. Good luck studying.